Hello, everyone, and welcome to today's webinar, Digital PCR Foundations. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Dr. Dave Bauer, qPCR and dPCR Field Application Scientist at Thermo Fisher Scientific. Dr. Bauer, you may now begin your presentation.
All right, thank you for the introduction. So we will be talking about digital PCR basics today. And a quick overview of what we're going through. We'll talk just in general what digital PCR is, some comparisons of digital PCR to real-time PCR, and also understanding the workflow for specifically the QuantStudio Absolute Q digital PCR instrument. And then we'll finish off with some common applications and areas where digital can be very valuable to use. What is digital PCR? It's a method to quantify nucleic acid targets without standard curves. So let me just pause that sentence right there. And if we just think about the beginning part of quantifying or even assessing nucleic acid targets, that ability has been around for a while, even going back to traditional or bulk PCR with the gel. In those cases, in lanes 2, 3, and 4, we can see that our target was present, and in lane 5, it was absent. And then the next advancement in PCR came with quantitative or real-time PCR. In this case, as the name real-time suggests, we're not just doing an endpoint measurement, but we're actually looking at the PCR reaction as a function of PCR cycles. So we get that whole curve showing the kinetics and efficiency of the amplification. And you might be thinking, I've done quote-unquote absolute quantification with real-time PCR. Keep in mind, though, real-time PCR is specifically a relative measure. The output we get, the CT value or cycle threshold value, it's a relative measure and generally needs to be compared to other CT values for meaning. And this is where the latter half of that sentence up top comes into play, where we can do this without standard curves. And that's accomplished by dividing the bulk reaction into many subreactions, and in this case, thousands and thousands of them. And in that case, when we've done our PCR cycling, we don't get any real-time information, but rather with digital PCR, it's more analogous to a gel where we get more of a binary yes, no, do I see amplification or not. All right, so let's look at some of the strengths of digital PCR. So for starters, as we mentioned, digital PCR, we can quantify our sample without the need for any reference or standard curves. Additionally, digital PCR is really great at detecting rare targets amongst a sea of wild type, often called the looking for the needle in a haystack problem. And there's also a linear response in digital PCR compared to the logarithmic response in signal with real-time PCR. And this makes detecting very small changes very helpful. So even if you're not doing absolute quantification, but just relative, such as gene expression or copy number variation, digital PCR can still be very beneficial there over real time. And we also have single molecule resolution. So not only can the instrument detect and quantify the presence of a single target, but another value is that we can also confirm that positive counts where a target is seen, we can actually find out if those counts are in the same physical microchamber or subreaction. So we can actually get a lot more information about not only is the sequence present in my sample, but is it present and also physically linked to the other targets I'm detecting. This opens up a much wider range of questions that we can tackle with digital PCR. As well as an improved tolerance to inhibitors. We'll talk about that point specifically and many of these points on this slide going forward. So just wanted to introduce them here as they will be kind of repeated themes as we go through. So let's do a little more deep dive on digital PCR compared to real-time. So they're both methods to assess or quantify nucleic acids, and they use really the same underlying biochemistry. So whatever we're using for real-time PCR, all of the master mix components, our template DNA, that's really going to be pretty much the same with digital PCR. The underlying biochemistry isn't different. However, it's the data collection is a big difference. We don't actually need amplification curves, so we're not dependent on reaction kinetics or the efficiency of the PCR reaction when we're talking about digital PCR, because it's just the endpoint detection. So let's dive a little bit deeper into the comparison of digital PCR in real time. So starting on the right of the slide, real-time PCR does have its areas where it can be beneficial compared to digital. So for example, developing assays, as I mentioned on the previous slide, that we're detecting with real-time PCR, the kinetics of the reaction, and that can sometimes be valuable for assay development. Or let's say we're just doing relative quantification, looking for a 10 or 50-fold change in gene expression, that might be sufficient to do real-time. Or if there's a large number of targets or a large number of samples that we're trying to assess. Additionally, if we're simply trying to confirm if a target is there or not, present or absent, in that case, real-time might be fully sufficient. One other big value of real-time PCR in the middle of the slide here is the dynamic range. Real-time PCR has a large working dynamic range where we can detect a handful of molecules, copies of our target, all the way up to millions or billions of copies of our target in the sample. The dynamic range of real-time is very beneficial for that. Digital PCR, it's going to be a smaller dynamic range, but it's more geared toward the lower concentrations. So anytime we're working with lower concentration targets, that's where digital is potentially going to be more likely to have greater value. And so looking at that list on the left, measuring rare targets, of course, and as we've alluded to multiple times, whenever absolute quantification is needed, that's where digital is going to shine. And because of that precision that we referenced, we have the ability to monitor small changes, whether that's over time or between samples. And multiplexing. We'll talk about later how multiplexing can be easier with digital compared to real-time, primarily because we don't rely on the reaction kinetics. And lastly, quantifying standards for real-time PCR. So you notice the bottom left of the slide and the top right, those are complementary things. I don't want you to walk away from this thinking, should I use digital or real-time? Sometimes the answer is going to be both. They do have kind of a yin and yang complement in certain workflows. So let's step back and just look at the data collection for digital PCR. And we'll start by looking at it for real time and then go from there for comparison. So as we alluded to earlier, the big difference is the efficiency of PCR. That is a huge aspect of real-time data collection. So that plot on the graph is showing if we had a sample where the efficiency was at 100%, that line would cross the threshold at a certain place, giving us a certain CT value. But if that same sample or that same concentration was in the presence of inhibitors, that line instead might be kind of less steep, and that's going to impact the CT value. That's where real-time PCR is very vulnerable to inhibitors for this reason. But with digital PCR, we're not looking at the progression of the reaction. We're not getting CT values. We're just making the binary distinction of, do I see signal or not? So basically, we're looking at the very end of that graph. As long as the blue line got above the baseline noise, that's a sufficient amount of amplification for digital PCR to do what it needs to. So let's look more at data generation for real-time PCR. So let's just zoom in on one well in our plate. So in this case, we have our bulk reaction. There might be one target molecule or tons and tons of them. But for each target molecule present, that one copy at cycle 1 is going to get amplified for cycle 2, and so on and so on. So that by cycle 40, we have tons of copies, tons of signal generated from that starting amplicon. And the real-time PCR output is an amplification curve. And so there is the progression of signal over the 40 cycles. But now let's say I wasn't trying to quantify anything at this point. I just wanted to say or ask, is my target there or not? Well, just looking at cycle 40 alone from the real-time data, we can see when there was target present, the signal went up. So at cycle 40, it was elevated compared to when there is no target present, its signal stays at baseline. So we can differentiate just from the endpoint measurement if target was there or not. So keeping that idea in mind, let's now switch to digital PCR. Instead of running the entire reaction in one bulk set, let's take the liquid illustrated by that purple box coming out of the H12 position on the plate, and then dividing that out into thousands and thousands of subreactions in that grid there. And these subreactions are physically isolated from one another. And so with that physical isolation, now if we have a particular subreaction that had target present, then after PCR thermal cycling, we're going to see signal in that particular location. But because we divided that sample up into so many reactions, they're going to be pretty small volumes, less than a nanoliter. At that point, we expect that some of those subreactions actually won't have any target present. And the subreactions that don't have target will not give us signal. And now if we look at actual data from the absolute Q instrument, this is what it would look like. The endpoint measurement, we see that for any of those isolated subreactions where we have some target present in the beginning, we get the fluorescence shown in green. And then for any subreactions that were empty of target, we don't see anything. And as you would intuitively assume, if you have a higher target concentration, you'll have more fluorescent reactions. Whereas if you have a lower target concentration, you'll see fewer fluorescent reactions. So this view here on the left is showing the full array from one instrument run. In this case, there are 20,480 microchambers or subreactions that the one sample got split up into. And so if we zoom into one little section, we can see the bright squares and the dark squares. And this information alone is what's used to get the absolute concentration of our target. So we typically look at this data as a scatter plot where that horizontal axis represents the 20,000 plus microchambers. The vertical axis is the fluorescence intensity. And what we want to see is a nice separation where we have high signal and then somewhere else low signal. And as long as we can differentiate positives from negatives, we can then get a concentration. So all we need from digital PCR is a count of how many of those subreactions were positive. In this example, 3,630 were positive out of 20,466. Those numbers alone get us to the concentration of 468 copies per microliter. And this is done relying on Poisson statistics. And the reason that we need to incorporate that is because we can't just naively assume that every fluorescent chamber corresponds to exactly one target molecule. That might not be the case, and that's where Poisson statistics can help. So just to kind of briefly give an example about Poisson statistics, let's talk about a quick example, a little hypothetical here. Let's say I have this grid of squares on the table, and I have a cup of red balls. I dump the red balls into the grid. And let's assume that we have the same number of red balls as we have squares in the grid. So the effective quote unquote concentration of one ball per square. But if I were to dump those out on the table, I don't think anyone expects to see exactly one ball in every single square. Instead, we know that there's going to be some randomness, that some squares will be empty, some will have one ball, two, etc. Poisson statistics can help because it tells us, knowing that average concentration of one ball per square, denoted by that lambda symbol, having an average of 1, using Poisson statistics, we can tell what that distribution of how many balls in each square would be. What percent would be 0 or have 1, 2, etc. And if we change that concentration, let's say I used twice as many balls per square, then we have a lambda or concentration of 2 on average. And in that case, there'd be a different distribution, and so on and so on. For any effective concentration, we would get a different distribution here. But the catch is, with digital PCR, we can't actually measure the whole distribution. All we can really see is if a spot was empty or if it was not empty, one or more. So that vertical dashed red line going through the graphs separates what we can actually see with a digital PCR measurement. So in reality, or in digital PCR, it would look more like this, where we can't tell how many balls are in the squares, but we can tell which squares are empty and not empty. And thankfully, with Poisson statistics, that is still enough information to get an estimate on the concentration. If we just know the proportion that are empty, in this example, 37% of the squares were empty, Poisson statistics gives us the equation to plug that number in. We plug in that 37%, that gives us 1 ball per square, or thinking back to digital PCR now, 1 copy per chamber. And now 1 copy per chamber still isn't the most meaningful measurement. We want something more like copies per microliter, which is an easy conversion as well. So just by knowing simply the percentage of microchambers that are empty, we can infer what the concentration was. So now let's talk about the digital PCR workflow, specifically as it relates to the absolute Q instrument. So preparing our samples is going to be a lot like real-time PCR, or even pretty close to traditional PCR with a gel. We're mixing up the components, the biochemistry, to make PCR happen. But with digital PCR, as we've talked about, we then need to have that sample divided into the thousands and thousands of sub-reactions. Then there is thermal PCR cycling, which then lets us see which of those spots are containing our target and which ones are not. And this is where the idea or the name digital comes from. We're separating out the sample so that we have tiny little mini versions of that sample, and then we're making that binary yes, no, or digital 1, 0 check to see which spots have our sample or our target and which ones do not. After the thermal cycling has occurred, some of those microchambers will be fluorescent. That information is captured. And then finally, those counts of positive and negative are made to get a concentration. And it's the way that the reaction is subdivided for digital PCR that really is one of the defining features for different instruments that are out there. The way we divide the sample is critical to all the steps that have to happen before as well as after. And it also critically impacts what level of data quality or data quality control metrics we can have in place, depending on what technology we're using to divide the sample up. For the QuantStudio Absolute Q instrument, the heart of that is the plate, the MAP16 or microfluidic plate with these 16 arrays. This contains a lot of the technology here that gives us the ability to separate that sample out into those individual thousands and thousands of microchambers. And with this MAP16 plate, this process is very repeatable. We get a consistent number of microchambers created, and this is also done completely hands-off from the user. So So that MAP16 plate, again, that's 16 arrays or 16 samples that we can look at per plate. But the instrument gives you the flexibility to actually run subsets of the plate at a time in intervals of 4. So 1 column, 2 column, 3 columns, or 4 columns can be run on a given experiment, which means you don't have to wait and batch your samples so that you run all 16 and then avoiding any wasted plate usage. In this case, you can run a partial plate, get some information, and then run the next plate, which is really helpful for either low throughput labs or when you're doing method development where you just want to change one variable, see its impact, and then try the next iteration. And inside of each of those 16 arrays, these images here are showing a zoom in, looking at the actual microfluidic system. So you can see for a given array, there are 20,480 fixed chambers. These chambers are physically defined by the geometry of the plate itself in the microfluidic system. And inside of that array will contain 9 microliters of our loaded PCR reaction mix. And what's important to notice is that we're not really wasting sample. Over 95% of that 9 microliters that we input is actually going to get utilized and assessed in our digital PCR reaction and in our data collection. So very little dead volume here. And part of that comes from the fact that we have such a consistent, successful microchamber fill rate. Microchambers we're going to be utilizing pretty close to 20,480 every time. And all of that combined gives us the confidence to know that we have precise, repeatable measurements, which is important for accuracy and consistency. So from the user's point of view, the way this process looks, they would take a standard pipette, add 9 microliters of the reaction mix into one of the loading ports. Then with a standard pipette again, add 15 microliters of a special isolation buffer. After those 2 simple strokes of a pipette, we can then place these rubber gaskets over the plate openings and place the plate into the instrument. And that really is all that's required of the user. It's a very hands-off process. From here, the instrument's going to take the plate, do all of the necessary separating into the subsamples using its special pressurized system to move the fluid through the microfluidics. It's also going to do thermal cycling with the heat block that's in there and capture the fluorescence data. All of that occurs without any further intervention from the user. So just some key takeaways about the Absolute Q instrument. Most of this list we've talked about. The ones I haven't hit on yet, the quick time to results. That whole process I just described is done within around 90 minutes. So you get data quickly. So if you do have a large number of samples you need to do, you can run many, many plates throughout the day. Also multiplexing. We'll talk shortly about this, how multiplexing gives you more information per run. The Absolute Q is a 5-color system. One of the colors is for quality check, and then the other 4 are available for target multiplexing. And the system has a unique ability for auto false positive rejection, which means we can have great confidence in our data, which is extremely important when you're dealing with low concentrations, where each positive spot, each fluorescent signal, we really need to know that it was truly because of our target and not other artifacts. Let's turn to some common digital PCR applications. So this slide is showing absolute quantification, rare target, copy number, gene expression, multiplexing, all places where digital PCR can be very valuable. And any of those applications can be applied to a range of research areas. We'll touch upon a few, but some common ones are cancer research, cell and gene therapy, as well as wastewater testing. So with digital PCR, one of its key strengths is the ability to absolutely quantify target. So let's talk about that a little bit more. And again, you heard me say this in the beginning. You might be thinking, I've either done or seen or heard of someone doing quote unquote absolute quantification with real-time PCR. While that can be done, keep in mind it was still a relative measure. So there are a couple caveats when we're doing absolute quantification with real-time PCR. One is the fact that we need this reference standards. So we need to have a range of concentrations of some reference target that is supposed to represent our sample. And sometimes it's not easily available to get reference material that is a good representation of your particular sample or target. And so it is important to know and keep in mind that you're making a relative measure from a sample type that's a reference to the sample type that's an unknown. And if there's something about those that are different, whether one contains maybe some inhibitors or there's something in the matrix itself, if that impacts the PCR efficiency, that's going to impact your quantification. As well as the accuracy. So in the graph there, you're seeing we have our references, those orange dots shown, but you need to know ahead of time from some other orthogonal technique, what was the concentration of your reference standard? And sometimes that's not always a reliable number, or it can be difficult to determine. And another consideration is repeatability. Because we're doing a relative measure, that relative being from the standard curve itself, any variability in the standard curve is going to impart variability in your unknown samples measurement. So different analysts, different users, different labs, different lots of references, all of this can impact how repeatable our measurements will be for a given unknown sample. And of course, workflow finally. So looking at the top right, the standard curves, you have to put those onto your plate. They consume time, resources, reagents, money, and you have to repeatedly set those up as good practice. So it just has another extra level of impact on the workflow. This slide's just summarizing everything we just went over with the drawbacks of standard curve. And now let's turn to digital PCR for absolute quantification. So if we're doing absolute quantification, generally the need is for some consistent, precise, and accurate quantification of our target. And for all those reasons we just mentioned, all those drawbacks with real-time PCR, digital gets around those because, again, it's a direct measurement. There's no standards that we're comparing to. We're directly assessing concentration. So one example of where you care about absolute quantification, let's say we have some viral vectors that we're developing and we need to quantify how much of our usable vector is present in our formulation. So this example here is showing a case where we're looking at the AAV, or adeno-associated virus, and quantifying it over a dilution series. So starting out with really high concentration, doing several tenfold dilutions, we can see that at the very high concentrations, the green clusters in the middle of the slide, most of them are positive at the highest concentration, and we see the progressive decrease as we go rightward to lower concentrations. And what's important here is that the plot in the upper right corner is showing that we have good linearity here. So these are just simple dilutions. We expect the concentrations to change accordingly, and that's indeed what we're seeing over these 4 orders of magnitude change in concentration. Digital PCR can be very effective at quantifying targets. So with digital PCR, we also have the ability to do multiplexing. As I alluded to this earlier, multiplexing means we can get a lot more information from a given run. So that multiplexing, you can use that for extra redundancy in your experiment. Maybe you have the same or different targets where you expect the same concentration, gives you more assurance in the measurement that we're getting out. Or if you want to build in some additional internal controls, whatever options you think of, they'll be beneficial to make the experiment better with multiplexing. Additionally, digital PCR can do more than just tell you the concentration of the different targets. It can also tell you if they're physically attached to one another, something that real-time PCR wouldn't be able to do. So for example, if I have some viral genome and I've picked 4 different locations represented by these 4 different colors, I would expect for intact molecules that where any microchambers that have blue signal would also have green, yellow, and red. And that's something our software allows you to assess as well. So you can confirm for molecular intactness of genomes, not just their presence or concentration. And that can be really helpful if you are building a viral vector where maybe you need to confirm that is the backbone sequence there, and then my insert, and then any other parts that should be. All right, so let's turn now to rare target detection. So digital PCR's strength is the ability to quantify at low concentrations. So real time might be able to still give you a more qualitative assessment, but with digital, we not only get the qualitative, but we're getting that quantitative information at very low concentrations. Digital PCR also has the precision to give you reliable and monitor reliable changes in concentration at those low concentrations. So some common uses where that might be important if we're doing single cell analysis, or if we're maybe trying to detect small residual amounts of something that shouldn't be there, like residual host cell DNA or other contaminants, where we need that low level detection. Or there's been a lot of applications of using wastewater surveillance with digital PCR. Maybe we can actually look at changes in the SARS-CoV-2 genome in wastewater to help get more information about what's happening at the community level. And in those cases, that target is going to be pretty dilute when we look at the overall wastewater system. Another use case is liquid biopsies for oncology research. So in this case, we might be looking at circulating tumor DNA that's in the blood. One of the values of using a liquid biopsy is just the ease of obtaining from the patient. That means you can get more frequent checks to get more information about relative changes. Something else that can be valuable with digital PCR here is when you're looking for rare targets, but not just rare targets that are going to be in a sea of very similar sequences. Digital PCR has a huge advantage in the fact that it subdivides the bulk sample, making an effective increase in sensitivity for any of those microchambers that do have the rare target present. So looking at an example of oncology research, looking at liquid biopsies. Again, one of the motivations for the value of a liquid biopsy is it's non-invasive. It also gives you a better picture of the tumor overall. So at the biological level, if you were to just take a solid piece of tumor, it might not be representative of the full homogeneity of that sample, but the liquid biopsy can contain little fragments from different places on the tumor, giving you a more fuller picture. And again, mentioning that the ability to detect a sequence that's very similar to the normal or wild type sequence. So a lot of times with these cancer markers, we might be looking at a single base pair change where the liquid biopsy is completely full loaded of the wild type or normal sequence. And we're trying to find the cases where there's that single base pair change, and that's what we're interested in. So having the ability to see that needle in the haystack is really important and valued with digital PCR. So here's an actual example where this was put to use. So in this case, there was a patient where they had cancer, and we can see in the graph to the right, there was developed an assay to use digital PCR to monitor a sequence that represents those tumor cells. And a drug was applied that wasn't very effective, and that could be quantified and monitored based on the digital PCR results. But then the second treatment, it was seen that had a great impact, a severe reduction in the tumor sequence concentration. And that information can be very useful to monitor treatment efficacy and also to look if there is progression to any further stages or reoccurrences of the cancer after therapy or after treatment. And so this plot is just showing, you can see the tumor marker sequence dropping down and then also remaining low. So if we're trying to get early indications of any tumor markers, it's important important to have that sensitivity because the lower we can detect, that means the earlier we are in the tumor progression, the more valuable that information can be. And Thermo Fisher actually offers a wide range of pre-designed and also wet lab tested liquid biopsy digital PCR assays. And these wet lab tested assays, they have the data showing that we can detect down to as low as 0.1% mutant allele frequency, or MAF. And so in these plots here, you can see on the log Y scale, we're getting down to just a handful of target molecule detections amongst the tens to hundreds of thousands of the wild type sequence. And all of those assays, they can be easily implemented into a workflow where we have the liquid biopsy sample collected, some kind of pre-processing to clean up the sample, extract DNA, and then that sample is run on the digital PCR system. And again, mentioning that the digital PCR system takes care of all the sub-steps of digital PCR, very hands-off from the user, and then outputs the information that we need in terms of those mutant allele frequencies. And so with that, a little reminder that we have a lot of information on our website. If we go to thermofisher.com/dPCR, you can look at all of the different aspects of the instrument, as well as related reagents and pre-designed assays that are available. And with that, I'll turn it over if there are any questions at this point.
Thank you, Dave, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. All right, let's go ahead and get started. Your first question is, you mentioned that the absolute Q can support up to 4 targets per reaction. Developing a successful 3 or 4-plex real-time PCR assay can be tricky. How does digital PCR compare to real-time PCR for multiplexing?
Yeah, so we talked on a couple slides about how the efficiency or the kinetics of the real-time assay are important when you're dealing with CT values for real-time PCR. And when you're multiplexing, if you've ever developed a high-level multiplex, you may have seen the difficulty of balancing those 4 different assays trying to work together in the same reaction without interfering with one another. And so a lot of times it can be a bit of an art into dialing all the knobs to the right value to get all the assays to work effectively together. And the reason that it's such a precise tuning is because, again, you don't want to interfere with the efficiency of any of those reactions for real time. But with digital PCR, we're not as concerned or dependent on the exact efficiency of the assay. If we multiplex some things together and the efficiency does get a bit compromised for one of the targets, as long as we're still getting enough signal to go beyond baseline and see the present versus absent microchambers, we can still get accurate quantification with digital PCR in that case.
Okay, great. Thank you. All right, your next question. Since the dynamic range of digital PCR is smaller than real-time PCR, how confident can we be that our measurements are within the dynamic range of the instrument?
Yeah, so that's an important factor. And I did mention that the dynamic range is smaller with digital compared to real-time PCR. And if you're on the low end of the dynamic range, it'll be pretty obvious because you're going to be getting a very low concentration. And if you're at the high end, as you get higher and higher in concentration, there's going to be more variability or more uncertainty in that concentration. And that's where researchers can often be concerned, is at the high end, where it might not be as intuitive if they're too high or not. But thankfully, the digital PCR software for the absolute Q utilizes Poisson statistics to not only get us a concentration, the copies per microliter, but also is able to use Poisson statistics to give us and calculate an uncertainty interval, the 95% confidence interval. And that's being determined on a sample per sample, measurement per measurement basis using the number of targets that were found in that experiment based on how many total microchambers there were. So for each concentration that's reported in the software, you'll get a confidence interval as well. And so then you can directly see in your measurements, if I'm getting an uncertainty that's simply larger than I would be comfortable with, you'll know that you can then just dilute that sample, run it again, because as you get to lower concentrations, your relative precision is going to be better. And that's also reminds me of something I commonly tell customers if they aren't quite sure about where the concentration is going to be of the sample they're dealing with and they're concerned with this issue. Instead of running technical replicates on digital PCR, I advise to run dilutional replicates. Take that sample and run it as is, and then also do a 10 or 100-fold dilution of it as well and run that as on the same experiment or even a different experiment. And then you'll get more information. And if one of those is outside of the dynamic range, the other one could be. And if they're both inside the dynamic range, you should see great agreement between them and get even more confidence in your measurement.
Fantastic, thank you. Your next question, how does the instrument perform the mentioned auto false positive rejection?
Yeah, the auto false positive rejection is important for any experiment, but it's really critical when we're talking about rare targets. Because again, with rare targets, if you're only expecting or commonly seeing 10 of those microchambers being lit up, you really want to know that all 10 of those are true positives, because 2 of them being false positives can have a huge impact on the relative difference in our answer. So the absolute Q system has actually 2 ways to help with that, with auto false positives. One is that the instrument actually takes 2 images. So there's the endpoint detection image that we've been talking about, but there's actually another image collection step really early on in PCR cycling to get the background fluorescence levels from that particular sample on that particular plate. And this is collected for every single individual experiment and then subtracted out from the endpoint image. So that means if there was any fluorescent artifact present, it would get subtracted away, meaning it would not contribute to a false positive because it would get subtracted out. And there's also another more sophisticated layer where the software isn't just looking at the overall intensity of fluorescence. It's also looking at the inside that image, the pixel by pixel intensity of fluorescence signal, that distribution of fluorescence intensity within the microchamber, and it knows that there's certain patterns that represent real PCR amplification versus perhaps an artifact. And so that's also used to make sure that things called as positive truly reflect PCR fluorescence, not artifacts.
All right, thank you, Dave. We've got time for a couple more questions. Your next question, do I need to redesign my existing real-time PCR assays or change how I typically design assays to use absolute Q?
Generally, no. The underlying biochemistry is really the same from digital and real-time PCR. So if you have PCR assays or real-time PCR assays that you've been using, those generally can be used with digital PCR with no optimization, no change. I've walked into plenty of customer labs and we just wanted to try their real-time assay that they've been working with. We run it on the absolute Q and the data is perfect. No need to optimize. And again, that's because the underlying biochemistry is pretty much the same. And in terms of developing any newer assays, really the guidelines that you would use for designing an assay with real-time would also apply to digital. So anything about the primer sequences, the lengths, the sequence context, all of that is pretty much going to be the same for digital as it was for real-time.
All right, great, thank you. And your last question today, what is dead volume and why is it important for digital PCR?
Yeah, we briefly alluded to the idea of dead volume, where let's say in a system, we're going to load 10 microliters of our reaction mix into the instrument or into the plate to get assessed. Some instruments might take, say, 10 microliters and then only actually utilize and get data from a fraction of that, maybe half that volume or something like that, because some of that volume maybe wasn't effectively incorporated into a subreaction. And if that subreaction isn't usable for one reason or another, and it doesn't make it to the final end of the data analysis, that volume from that subreaction is considered dead volume. Or any other volume that never makes it into any subreaction at all, dead volume. With the absolute Q system, we talked about that there's over 95% of the volume is truly utilized and actually gets data associated with it. So that's less than 5% of that being dead volume. And that's important for two reasons. One is reagents. They cost money. That dead volume consists of your assay mix and your master mix. So that's money going away. Secondly, if you're looking at rare targets, if you're only assessing 50% of what you loaded, you're going to have a lot more uncertainty, a lot more variability in if I detected my target. Was it in that 10 microliters, but not in the 5 microliters assessed? Now you just have another level of variability, another subsampling issue to keep in track of. Whereas with our system, we're having over 95% of that volume utilized, so we don't have that concern.
Well, thank you again, Dave, for your time today and your wonderful presentation. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.